And coming back to the structure of DNA, you get to these proteins, they're getting the DNA wrapped up on it. So how do you remember what they are? First thing you associate, you look at them and you say, oh, they are little balls, and it has to do something, okay, not a hysterectomy, it's a histone, okay? Histones. Histones, those are little balls associated with DNA. Those stones that are in the core here, yes, this is important, it's going to be in the test. Yes, hello. Those stones here, the histones, they are in the middle here, they are 2A, histone 2B, okay, those are 2, and then you have histone 3 and histone 4, and then you just get 2 of these here, 2 of those, 2 of these, 2 of those. That will give you a 4 little balls structure. That's your histone core. So that's your nucleosome core. So these are the histones that are part of that one. Now, where is histone 1? It's not there. Okay. Histone 1 has to be different. It's not in the core. So when you look at the DNA that's going to get wrapped around these histones, first, how does it get wrapped around? It has to have some kind of attraction, right? So you know DNA is negatively charged, so these histones must have some type of positive charge to attract your DNA. Okay, so they usually have a lot of lysine residues that are positively charged. DNA gas here goes around these histones, usually about 2 turns, maybe a little bit more than 2 turns, right? And then for this whole thing to be held together, that's when histone 1 comes along. Histone 1 is this little strip-looking thing here. So histone 1 keeps this core together, and now this core with the histones 2, 3, and 4, and the DNA is called the nucleosome, and then if you add the histone 1 to it, you get this chromatosome structure. Then you know, now in between one chromatosome and the next, you have this DNA that is not protected by anything. Okay, so that's the linker DNA. That's the naked DNA. Okay. If you hit those, they are really going to suffer. Here you look how these histones are going to get together, and then they're going to pile up on top of each other and get you more condensed structures here. Okay, you get more condensed, and then they start turning around and compacting. Every time you compact a little more and more DNA into a small amount of space, and then you finally get your chromosome. Now, looking at these histones, these nucleosomes structures, as I said, you have this piece in the middle. It's called a linker DNA. It's linking one chromatosome to the next. And what happens here, since I mentioned this part here of the DNA, since it is naked, is easy to attack. So if you add some nucleases in there, the nucleases are not going to reach the piece of DNA that is protected by protein. It's going to reach the one that is easy to attack first. Remember, the cord always breaks at the weakest spot? We have DNA breaking at the linker DNA. Now, when you look at the structure of the chromatin, sometimes you find these little puffs. What do they really mean? DNA is all very much organized, but look at it. If you look at this, and this is all the DNA, okay? Non-coding DNA, coding DNA, genes, junk, everything is stuck on these histones. But the idea is that you have to get from DNA to protein. You have to transcribe the DNA and get into RNA, and then push it out to the cytoplasm to get your protein. How are you going to do this if you have this very compacted structure that is basically non-penetrable? You have to figure out how this thing is going to open up. So we're going to go into more details of this, but remember, histones are positively charged, and then DNA is negatively charged. You're going to have to change some of these charges in order to separate these guys here. They're pretty much stuck together if they're positive and negatively charged. Okay. When you start to separate your DNA from your protein core, the DNA will get relaxed, and the histone is going to be on the side, so you can transcribe or you can replicate this DNA. And what you see are these chromosomal puffs here. Doesn't look very organized, but it looks like a little ball that is in the middle. Now, when you see this in the microscope, you know that these are places of high transcription. Okay, if the cell is in the is in G1 stage, is not going mitosis, not DNA synthesis, and you see these puffs, you know that they are sites of high level of transcription. So some gene is going to be transcribed out of this puff. Now we also know that at the end of the DNA, there is nothing else, right? You have to have an end to the DNA. It's not a circular DNA. It doesn't go on forever like in the bacteria. So what happens at the tip of the DNA, at the very, very end of the DNA? Now this helix is going to be going on forever, but it's going to have to end. But if you have naked chromosome at the end of the DNA, it's going to be very vulnerable to nucleases. Anything could attack those ends and chop up your DNA from the end to the center. So in order to protect that end of DNA, there is a specific structure that this DNA forms. So you don't have to know very much the specifics and the sequences of this, but just keep in mind that in order to protect the ends, this DNA is going to pretend that it's circular, when it's really not. It's a linear DNA. It's not a bacterial DNA, but in order to protect itself from nucleases, it's going to behave like it is a circular DNA. So what it does, it folds on itself, it finds a matching spot that is close to the end, and just interacts with it, and you see it kind of looks like circular. So these are the structures at the telomeres, at the very ends of the chromosomes. Now, just a little thing about these transposable elements. These are DNA sequences, they are jumping all the time. So as I mentioned here, up to 45% of your DNA is composed of jumping sequences. You just picture in your head this nucleus with almost 50% of this DNA behaving like jumping beans. I think it's a mess, but it's really not. Okay, it's very organized, and we are proof that it's very organized and very efficient. We're not really mutating at every generation. It's just important to keep this in mind, that this happens. Now, how do you recognize when you have this transposable element or when you don't have it? The way you recognize it is by looking at the sequence of DNA, and then you find something that is exactly the same as the sequence on the other side of this thing that got inserted in the middle. Now, how does that happen? There are proteins, there are nucleases that are going to cut this DNA here in order for a jumping DNA, transposable element to be connected to that side. Once you have a cutting here you have one sequence, and this has a different sequence here. But when this blunt end piece of transposable element comes in the middle, what happens here? There is a gap. There's a gap that has to be filled. So then polymerase will go there and it's going to fill the gap. Now, because we know that it's going to make the exact match from the other strand, they end up to be the same on both sides of this transposable element. So this is how we can map out where transposable elements ended up. So this is as much as you have to know for chapter 11. Now we go into the chapter 12 of replication and recombination. There are certain characteristics that DNA must have, that the inherited material must have. It has to be copied accurately. We don't want to be mutating at every generation. It has also to replicate semi-conservative manner, which we will go into a lot of details there. And then we're going to see how exactly replication is taking place. How do we know that DNA has to replicate? Well, if it did not replicate and the cell divides, what's going to happen to the DNA? If you have no replication, you have 46 chromosomes here, and then you divide the cell into two. That means no replication, you're just going to divide your 46 into two cells. You're going to have 22 and 23. And then if you divide these cells again, you're going to lose some more DNA. You don't replicate them, so they're going to go down and down and down. After about 10 divisions, you end up without any DNA left. That's not a good idea. In order to maintain the correct amount of DNA, you just have to replicate it every time the cell divides. which is pretty much common sense. How does that happen? You have all these chromosomes, about 6 billion base pairs, and if you think about the size of this, if you type one letter for each base, you can come up with 1,200 of your textbooks. The big one, the genetics textbook. Put 1,200 textbooks in one room. That's the amount of DNA you have in one cell. If you use font 10 and you type every base into a piece of paper, this is a lot of DNA. This is a lot of books. So when you have to replicate this DNA, you can assume that you might have a lot of errors. These are proteins doing the job. So you can assume there are going to be a lot of errors to replicate all these 1,200 textbooks into 1,200 textbooks more. But then, do you think you have lots of errors that are occurring during replication? Yes, you have some errors, but not that many. The DNA is very accurate when you replicate it. So usually you have maybe one error for every 10 million nucleotides, which that's a very, very small chance of error. So how is it that the DNA is replicating? Well, before the scientists knew that DNA was replicating in a somewhat conservative way, they had 3 ideas in mind. They said, okay, we have a double helix, and then the DNA can replicate in a conservative manner. And it replicates in a conservative manner. First thing you have to do is replicating. Okay, so from one set is going to become 2 sets. If it is replicating in a conservative manner, this means that the original DNA is going to give rise to another DNA. So here's the original DNA. It's going to give rise to another DNA. And then this DNA is going to be for the new cell, and this DNA is going to be for the old cell. So the old cell always keeps the old DNA. This is a conservative manner. Okay? And the other idea, so this is the first for conservative. The other idea was that you could have a semi-conservative replication. So in this case, here's the original DNA. It's going to replicate, and then each daughter cell is going to have a piece of newly synthesized DNA and a piece of the old DNA in there. So you have old DNA and new DNA. This is one cell, this is the next cell, 2 daughter cells. This is semi-conservative matter. And then you have this dispersive or random matter, which I don't know who came up with that idea, but here you have dispersive, in which here you have your original DNA, and then it's going to replicate, and now you're going to have your daughter cells. So each one is going to get some piece of DNA here and there, and somehow you're going to mix them all up, and you're going to find each other and know exactly what the original sequence was, and voila, magic, you get 2 daughter cells that are all mixed part of DNA, new and old, and every strain, in every strand. That is the dispersive manner. I think it takes a lot of imagination to imagine how that is going to replicate. And thank goodness it's not what's happening, because can you imagine to study all the stuff that will happen there? So let's see how they figured out that DNA was replicating in a semi-conservative way here. We have Melselssohn and Stahl experiment with the isotopes. This is when they use not radioactive isotope, but they use a heavy isotope here. They are going to be making DNA using fat isotopes, heavy. Okay, so they can separate it from the newly synthesized DNA. Now, how do they do that? They get a culture, bacterial culture, and they use this heavy isotope, nitrogen-15. So they have a soup here full of bacteria growing with nitrogen-15. Okay, they're going to be heavy bacteria. Okay. Then after they're growing nice and happy, he decides that he wants to see what's going to happen if he puts now isolated bacteria, spin them down, get a little pellet. Now you put in a broth that has just regular nitrogen, not heavy anymore. Okay, so now what's going to happen here you're going to have the DNA, you know, nitrogen is going to be nitrogenous base, is going to be marking the DNA, right? Not the protein. So now you are going to see what's going to happen to the DNA here. The DNA that is coming from this side is all heavy, because after a lot of replication and a lot of cell division, you basically have a lot of heavy nitrogen incorporated into all these bacteria here. So to get the bacteria put in this new broth, there is no heavy, no, it's just only normal. So from here, you're going to have some of the DNA that came from there, and some of the DNA that's going to be made once the cells divide. So now they're going to look at this and say, let me see what this DNA looks like in order to figure out how the replication is taking place. So it says, for the first cell division, after I add from this to that, actually, let's look at this one first. If I get all these bacteria and I put in a density gradient centrifugation, you know which one this one is? Density gradient has to do with the buoyancy of it. So we put in the tube called density centrifugation, and this DNA here is heavy. It's going to show up over there somewhere. Now, heavy means goes down the buoyancy. Have a lot of muscle mass, you sink in the pool. So this is how this is going to look like. Now, if you have a first cell division and you look at this DNA, it can tell you a lot about what happened to the DNA replication. Because now the hypothesis is that if you have a conservative way of replicating this, after one cell division, you should have your heavy original DNA here at the bottom, and you should have your newly synthesized DNA somewhere here because it's lighter, right? So this is the regular DNA, the regular nitrogen-containing DNA, and this one is your heavy DNA. So after one cell division, you get two bands, one heavy, one light, conservative. Now, if you have semi-conservative, what's going to happen here after one cell division? You have one band, because now you're going to have this DNA, it's going to be more or less in the middle of these two here. Middle in density, but you have one DNA that is composed of the two of them. It's going to have some purple. It's going to show up right there, both of them. You're going to have one band if it is semi-conservative. Now, if this is semi-conservative, now let's see what happens if you have the dispersive method, right? Dispersive method here, what's going to show up there? One band as well. Okay. Just like the semi-conservative, one band, no problem. This is first replication. Now if you have another replication, another round of replication, so you have all these cells that are going to divide again in a non-heavy nitrogen media. So what's going to happen with the pattern of the bands here? If you have conservative, you are going to get what? You're going to get the heavy band and you're going to get the light band. Same way as you had before. It's conservative, it's keeping the same way all the time. Now, if you have semi-conservative, what you're going to get? Well, now, if you look at the DNA here, this is the first cell division, right? If you divide this again using nitrogen that is not heavy, let's see what happens to this DNA here. It will become, once you make new DNA, the new DNA from this side here is going to be like that. And then the old DNA is going to be like this. One for the old one and one for the new one. There you go. So after the second cell division, you're going to have two different types of DNA. One that is going to be a mix, that is still going to show up in the middle here. But then you're going to have this new type of DNA that is only coming up from this strand and is using the non-heavy isotope. So now you come up with a lighter band that shows up over here.
And this is the second replication?
This is the second replication. Yes, so in the first replication, you had here from the original DNA, you had one that came up here, replicated here, replicated there. You have two DNAs coming out of here. Now for the second one, this is one of the DNA of the bacteria, you're going to have still semi-conservative this way, but now this part of the DNA here that is going to replicate as well is going to give you a DNA that is only made of the light isotope. It does not contain the heavy isotope anymore. So when you run it on this gradient centrifugation, you're going to get two bands again. So if that, two bands, why should you get four? You are talking about number of molecules of DNA, but I'm talking about where they float. They're all going to float. Molecules of one certain density is all going to float in one specific band. Heavy one is going to go down. But now the heavy one was only for the first replication. There's no heavy one there anymore. There's only the one that came from the first replication when the original DNA was taken. Okay? So here, yes, you have a lot more DNA. So I see what you mean. This band here, on the second replication, I mean, it's going to be thicker, not heavier, thicker. Okay? You're going to have more DNA here than you have there. Double, actually. I see what your point. More DNA here than there. Double the amount. Okay? But you're still going to have two bands because one of them is going to be the DNA that was the old and the new for the second replication. And then the other DNA is going to be only the light one because there is no heavy isotope there to make new DNA. Okay, this concept, I think at first is a little hard to get. But if you just use models, if you have just yarns, do the experiment yourself. Put yarns, cut some yarns, same length, put on the table, and you say, this is my first one, the red one is the heavy one, and then the other ones are going to be the new DNA. Do your first cell division and see how your DNA is going to look like. Your second cell division and see how the DNA is going to look like. And so the first one is always going to be a part of the old and then a new one. And then when it splits again, it's going to be still, one of them is going to be the old, there's going to be the replication, and then you're going to have two of the new ones from this guy here. Okay, if you have just the visualization of what is happening to your DNA replication, it's a lot easier to understand how these bands are showing up. Okay, so this is how they figured out that DNA was in a semi-conservative way. With the density gradient, there you go for the bands. If you have a semi-conservative, the first replication you have something that shows up in the middle. And if you have a conservative, you have them always separated. And then also if it is a dispersed form, it's going to show always the one band in the middle. It's always dispersed, so it's always one band in the middle. Because everything gets mixed up, and then you have replication, everything is still always mixed up. It's that thing that is hard to imagine. Okay. Yes, it's going to be a little higher if it is dispersed, you're right. It's going to go creeping up a little bit with each cell division. It's going to get lighter and lighter, but everything is going to be only one band. That's right. Okay. So with the second cell division is how they figured out that it was not dispersive. They knew from the first cell division was not conservative, and then they knew for the second cell division that was not dispersive. Anyways. So here we have the semi-conservative DNA replication. The strings first separate, and then you have newly synthesized DNA that will just get together with the other side, with the base pairing with the other side of the original DNA. And they use this heavy isotope, nitrogen 15. What's going to happen if he puts now isolated bacteria, spin them down, get a little pellet, now you put in a broth that has just regular nitrogen. So now what's going to happen here, you're going to have the DNA, nitrogen is going to be nitrogenous base, it's going to be marking the DNA, right, not the protein. So now they're going to look at this and say, let me see what this DNA looks like, and then put in a density gradient centrifugation. And this DNA here is heavy, it's going to show up over there, somewhere. No, heavy means goes down the buoyancy. So this is how this is going to look like. Now, if you have a first cell division and you look at this DNA, it can tell you a lot about what happened to the DNA replication. Because now the hypothesis is that if you have a conservative way of replicating this, after one cell division, you should have your heavy original DNA here at the bottom, and you should have your newly synthesized DNA somewhere here because it's lighter, right? So this is the regular DNA, the regular, just like the semi-conservative, one band. No problem. This is first replication. Now if you have another replication, another round of replication, so you have all these cells that are going to divide again in a non-heavy nitrogen media. So what's going to happen with the pattern of the bands here? Conservative, you are going to get what? You're going to get the heavy band and you're going to get the light band. Same way as you had before. It's conservative, it's keeping the same way all the time. Now, if you have semi-conservative, what you're going to get? So after the second cell division, you're going to have two different types of DNA. One that is going to be a mix, that is still going to show up in the middle here. But then you're going to have this new type of DNA that is only coming up from this strand and it's using the non-heavy isotope. So now you come up with a lighter band that shows up over here. But I'm talking about where they float. They're all going to float. Molecules of one certain density is all going to float in one specific band.